The PI3K AKT mTOR pathway is a master regulator of cell fate, controlling whether cell divides, becomes senescent, or undergoes apoptosis. The pathway is initiated by binding of a growth factor like EGF to receptor tyrosine kinase like EGFR on the cell surface. Binding to the AKT receptor promotes dimerization and relays the signal inside the cell by bringing the intracellular domains into contact, which causes them to undergo trans-phosphorylation.
This conformational change recruits target proteins that have an SH2 domain, including adapter proteins such as IRS and GLB2, as well as phosphatidylinoster 3 kinase or PI3K. PI3K is a heterodimer consisting of two subunits. Binding of the P85 regulatory subunit to RTK activates the P110 catalytic subunit. The P110 catalytic subunit contains a lipid kinase that phosphorylates the membrane-bound phospholipid sugar, phosphatidylinoster, converting it from PIP2 into the active form PIP3. PIP3 recruits proteins containing plextorin homology domain to the plasma membrane, including factors such as AKT and PDK1.
AKT is a potent regulator with over 100 targets. Its activation requires phosphorylation at two distinct sites. First, PIP3 activates PDK1, which phosphorylates AKT at threonine 308. Second, mTOR-C2 phosphorylates AKT at serine 473. Instead of activating the target, phosphorylation may also inhibit the activity of its target protein. AKT phosphorylates TSC2, causing it to relax its inhibition of LEP, which in turn releases mTOR-C1.
mTOR-C1 promotes protein synthesis by activating SCK1, a ribosomal subunit. mTOR-C1 also inhibits 4EBP1, causing it to release EIF4E, which promotes translation by binding to the 5' cap of mRNA. mTOR-C1 also promotes fatty acid synthesis by activating SREBP1, a master regulator of lipogenesis. AKT also protects the cell against apoptosis by deactivating pro-apoptotic factors.
Phosphorylation causes FOXO to be ejected from the nucleus and sequences in the cytoplasm, leading to downregulation of pro-apoptotic factors. AKT activates MDM2, which ubiquitinates P53 and marks it for destruction, preventing the guardian of the genome from triggering apoptosis in response to DNA damage. Phosphorylation of BAT releases inhibition of anti-apoptotic factors such as BCL2, which block the releases of cytochrome C from mitochondria.
AKT also upregulates XIAP, which blocks the caspase cascade by directly binding to initiator and effector caspases 3, 7, and 9. The PI3K AKT mTOR pathway is tightly regulated, and the most important negative regulator is P10. P10 dephosphorylates PIP3 and converts it back into the inactive form PIP2. mTOR C1, S6K, and GRAP10 also form a negative feedback loop that helps to attenuate RTK signaling and reset the pathway.
However, the PI3K AKT mTOR pathway affects almost every aspect of cell fate and is dysregulated in half of all tumors. It is often implicated in glioblastoma, endometrial cancer, breast cancer, and prostate cancer. Tumors also exploit the PI3K AKT mTOR pathway to promote metastasis via the epithelial mesenchymal transition. Pathogenic variants leading to sustained activation of the PI3K AKT mTOR pathway include loss of P10, amplification of AKT, and PI3K variants such as H1047R, which causes constitutive activation of the catalytic subunit of PI3K, even in the absence of RTK signaling.
Several molecular target therapies in this pathway have been approved, including PI3K inhibitors such as abrasive, compulsive, and diversive, AKT inhibitors such as capivasertib, and mTOR inhibitors such as everolimus and teneligliptin. However, resistance to these drugs often develops due to secondary mutations and activation of compensatory pathways. Combination therapies and biomarker surveillance can help block the escape routes available to cancer, but control of this critical PI3K, AKT, mTOR pathway is difficult to regain once it is lost.